All right, so now let's talk a little bit about DNA structure. We know that Watson and Crick determined the structure of DNA. We know that it's made of two strands of nucleotides that are then twisted together in a double helix. Each strand is built when the phosphate groups of nucleotides form covalent bonds with the deoxyribose nucleotides of adjacent, or the deoxyribose molecule of an adjacent nucleotide. Those are covalent bonds. Those are very strong. The bonds form when one deoxyribose attaches by connecting its 5' carbon to the 3' carbon of the next sugar in the next nucleotide using the phosphate group. The alternating sugars and phosphates make up what we call the backbone of the DNA molecule. The two strands are actually connected together by hydrogen bonds between the complementary nitrogenous bases. An A on one strand binds with the T on the other strand, and a G on one strand binds with the C on the other. The AT pairs are held together by 2 hydrogen bonds, while the GC pairs are held together by 3 hydrogen bonds. And that means it's a little easier to break apart the AT pairs than it is the GC pairs. They're held together a little bit more strongly. It's also important to remember that hydrogen bonds are these weak intermolecular interactions that occur between polar molecules. They're very weak compared to covalent and ionic bonds. It's important because we need to be able to separate the two strands of the DNA during both replication and transcription. So those bonds are breakable. So here's our DNA double helix. Here we can see the alternating sugar phosphates of the backbone, and then in the middle, we can see the nitrogenous bases that are connected together by hydrogen bonds. And again, here we can see 2 hydrogen bonds holding an AT pair together and 3 holding a GC pair together. And then here's a better look at sugar, phosphate, sugar, phosphate, sugar, phosphate. Also remember that one of these units, a deoxyribose connected to a phosphate, connected to a nitrogenous base, that is one nucleotide. And a DNA molecule is made of 2 long chains of those all hooked together. So each strand of the DNA molecule has 2 slightly different ends. One end is called the 5' end, and it terminates with an unbound 5' carbon on the last nucleotide, the last sugar of the last nucleotide. The other end, called the 3' end, terminates with an unbound 3' carbon on the last deoxyribose molecule. When the 2 strands are connected together, they're oriented in opposite directions. One of the strands runs from 5' to 3'. The other strand runs from 5' up here to 3' down here. Because they run parallel but in opposite directions, we say that the 2 strands are anti-parallel. Here we can see that maybe a little bit more closely. So this is the 5' end because here's carbon number 5. Down here, this is the 3' end because you notice that the 3' carbon isn't attached to another nucleotide. Over here, this is the 5' end because this is a 5' carbon in the sugar. It's not attached to another nucleotide. Here's the 3' end because the 3' carbon is not attached to another nucleotide. So this strand runs from 3 to 5 in this direction, and this strand runs from 3 to 5 in that direction. Parallel, but running in opposite directions. All right, so let's talk a bit about DNA replication. We know that it's when DNA is copied. The entire length of every chromosome is replicated during the process. It happens in the S phase of interphase. It's when a cell is getting ready to do either mitosis or meiosis. There's 3 main steps involved: initiation, elongation, and termination. Replication starts at these areas on a chromosome called the origins of replication. Each eukaryotic chromosome has multiple origins on each chromosome, while a prokaryotic chromosome has a single origin of replication. And it can do that because it's really small and it doesn't take long to replicate it. but the eukaryotic chromosomes are large, and if the process only started at one place, it would take a long time for this to happen. So here we can see the cell cycle, and remember that replication happens right in here during the S phase of interphase, the synthesis phase. Here we see that the bacterial replication starts at one origin of replication. It proceeds in both directions until ultimately we get two identical circular chromosomes. Here we see that in eukaryotes, the process starts at multiple origins of replication, proceeds in both directions until ultimately we get two identical copied linear chromosomes. So let's talk a bit about the initiation of replication. Replication begins when helicase, an enzyme, begins to unwind, or oftentimes we say unzip, the two strands of DNA at the origin of replication. This creates these little replication bubbles or replication forks. During replication, both strands of the DNA act as templates for the building of new DNA strands. These proteins, called single-stranded binding proteins, come in and attached to the unzipped DNA to help keep it separated and to stabilize them. Remember that this is starting at multiple origins, so we'll get multiple replication bubbles or forks on each DNA strand. Next, an enzyme called primase comes in and synthesizes a short piece of RNA called a primer. These serve as actual starting points of DNA synthesis. The primers are critical because the main enzyme of replication, DNA polymerase, can only add nucleotides to an existing chain. It can't start a new strand from scratch. So the RNA acts as the beginning of that chain. Think of it like a foundation for DNA synthesis. After replication is over, the primers are removed and they're replaced with actual DNA nucleotides. So here we see replication getting started. We have helicase unzipping the DNA. We have, it doesn't show it here, but we have some single-stranded binding proteins that attach and keep the two strands stable and keep them from reattaching. We have primase that will come in and build an RNA primer. On the leading strand, only one primer is needed, because this leading strand, the strand that's being built right here, it's going to be built from 5' to 3'. DNA polymerase, the enzyme that actually builds DNA, can only add nucleotides in that direction, 5 to 3. So this strand is built in one continuous piece. This other strand down here, the lagging strand, the new strand is going to have to eventually run from 3 to 5. Well, DNA polymerase can't build it that way. So as the original two strands unzip, primase is going to put down a primer, and then polymerase is going to build from 5 in this direction to 3 back in this direction. Well, by the time that fragment gets built, more is unzipped. So primase will come back and put a new primer. Polymerase will build from 5 back toward 3. And that continues to happen. So this lagging strand is getting built in little chunks. And these little chunks are called Okazaki fragments. Eventually, those chunks are going to get put together, and there's an enzyme called ligase, which will actually connect them into one continuous piece. Remember, though, that DNA polymerase, the main enzyme that builds DNA, can only assemble nucleotides in the 5' to 3' direction. So eventually, we get to termination. And by the way, adding those nucleotides, that's called elongation. That's step 2 of replication. And it happens as nucleotides are added in the 5' to 3' direction by DNA polymerase. Termination happens when two replication forks meet up on the same stretch of DNA. The forks converge. All the DNA is unwound. All the intervening DNA is unwound. Remaining gaps are filled and ligated or reconnected. And the replication proteins like the single-stranded binding proteins, are removed, unloaded. So oftentimes DNA replication is said to be semi-conservative. Both of the two original DNA strands act as a template for building new strands. So that means that the two new chromosomes that you get, like so, each one of these two pieces of DNA consists of one original strand shown in blue and one new strand shown in red. So that's why it's semi-conservative. Semi means half. So each new piece of DNA is half old, half new. And conservative because that old strand acts as a template. And it helps to make sure that the new strand has the right sequence. It helps it to avoid having mutations. So semi-conservative replication. As part of replication, there's also some built-in proofreading and error correction. DNA polymerase will go back and essentially check its work to make sure that the right sequence of nucleotides is there. If it detects a wrong pairing, it will remove it and replace it right away before continuing. This is carried out by DNA polymerase 3 as the new DNA is being made. A lot of the errors, most of them probably are corrected this way, but there might be a few that get by. So there is this process called mismatch repair that happens after a new DNA molecule is made. And it's its job to remove and replace mispaired bases. It happens when a protein complex recognizes the mispairing. A second protein complex comes in and cuts out the mismatched area. And then DNA polymerase will come in and replace the cut out section with the correct nucleotides. Finally, DNA ligase will seal it all together. All right, the last thing we'll mention here on the DNA structure and replication video are telomeres. So telomeres are these repetitive regions located at the end of each chromosome. They protect the actual genes located at the chromosomes from deterioration. Most of the time, they consist of repeated units of TTAGGG nucleotides. They're non-coding sequences. Really, they first came to light in about 1971. And what we found is that the very tip of a linear chromosome can't be copied during replication. So every time a chromosome gets copied, the tips shorten. If there were genes located at those tips, those genes would very quickly be lost or messed up. So the telomeres are basically throwaway pieces of DNA that can be lost without affecting the actual genes contained on the chromosome. There are a lot of scientists who think that the length of the telomeres helps to determine the process of aging. And eventually, when those telomeres get to a certain shortness value, they signal to the cell that it can't replicate anymore and that it should essentially self-destruct. It should go through the process of apoptosis. So scientists are really thinking that telomeres are important in the regulation of the cell cycle and also in aging and ultimately cell death. So there is an enzyme called telomerase which can lengthen telomeres. And this happens during fetal development. Once the fetus is developed, though, that enzyme shuts off and it degrades. And it's going to be inactive for the rest of the individual's life. So after fetal development, your telomeres continually get shorter and shorter. The only time that enzyme might become active again during an individual's life is if they get cancer. Some cancer cells have active telomerase. And what this essentially means is that those cancer cells can continue to divide very quickly and essentially forever. And they become immortal because their cells don't age. All right, we're going to stop there on the DNA structure and replication screencast.